Let me start by welcoming everybody from around the world. I do know that there are people from India. I know I just saw somebody from Sicily. I don't think the Australasians are with us because of the time zone, but I know we've got some Europeans other than Sicily with us. So I think this will be a very interesting opportunity. I have now been in the chair of leadership, professional leadership, for exactly 3 months. Started on the 1st of January. Lale Welsh, who was the previous CEO, left the organization on the 31st of December, and here I am starting the year. I wanted this to be an interactive session in which you learn a lot more about NDF, what we've done in the past, what we're doing today, and what we plan to do in the future. We have 3 excellent resources. Dr. Angela Leck, she will be doing a 6-minute video presentation. She will be joining us in person later for the Q&A. Dr. Weisleder is also going to be live with us, and Rich Horgan is also going to be live with us. Yes, every now and again you'll hear me say, next slide, please, Rachel. So Rachel, next slide, please. So the organization was founded in 2005 by Gila Michael. And the reason that it was founded was that she found that the daughter was diagnosed. She's still around, she's doing fine. But that was the impetus that started the organization. So the organization up to now has really focused on GNEM myopathy. But the Neurological Disease Foundation, by its name, I feel has a much wider outreach. And I think we need to expand our reach. By expanding our reach, we'll help others with other rare diseases so that we actually can be a real benefit to the worldwide community. And that's the plan. We're very grateful to her for starting the operation off. Rachel, next slide. We're very fortunate in having a very strong board. The top 4 people, Ralph, Tricia, David, and Carolyn, are the officers of the foundation. They serve on the executive committee. Ralph, as you can see, is a lawyer. David is financial. Tricia is an excellent resource and is very knowledgeable in the field. And Carolyn has basically been everything to the organization that ever there was. She was an executive director. She was the head of the board. She's now officially the secretary, and she's a wonderful resource to us. And we're really grateful. The people on the next 2 lines are, again, great resources. We're very fortunate to have them. Dr. Tahseen Mozaffar on the right actually runs a program for us at UC Irvine to help patients, and that's, I think, unique in the world. We're trying to expand it to global, but we're going through the process. At the moment, I know that for the next session in June, I think there's a patient flying in from India. So that's a big outreach. Sean Arianapur is the latest person to join the board. Sean has been an ambassador, and you'll hear more about the ambassadors in a moment, for many, many years. He sort of timed out of being an ambassador. It's a quasi-aging thing. He's been around with them for about 10 years. And we saw him as a wonderful candidate for the board, and he joined us at the last board meeting, which was in March. David Havarim has been an amazing resource to the organization for many years. Jason, again, a former ambassador, and we're delighted to join to have Tara and Amy join. Tara is a patient, Amy is a patient, and the way this works is Tara is the primary on the board, and if for any reason she's not able to attend board meetings or is just not available, then Amy will step in as the alternate. So very strong board, good people. They're a delight to work with. It really is. I'm very fortunate to come to an organization that has all this. While we're here, most people don't know much about me, which is excellent. I have a deep background. I'm a lawyer. I practiced law in England and then in Australia. I then got into the world of business. I was dare I say it, quite successful in Australia. I moved from Australia to England. I'm sorry, to America, to Los Angeles. Climate had something to do with it. I wasn't going back to the bad weather of England, as those in the UK know every day of their lives. And I got deeply involved in the world of nonprofit. I'm Jewish. I served as president of a very large Reform congregation in Los Angeles, 900 families. That congregation now merged with an even larger congregation in Los Angeles called Wilshire Boulevard. And the whole temple group is now about 2,500 members. I've served with many organizations worldwide, Save the Children Fund. I've deeply been involved in Israel and raising money for Israeli causes. And as importantly, I've been very involved in Jewish and non-Jewish operations here in Los Angeles. I served on the board of the Jewish Federation. I did an outreach program to newly arrived Iranian and Russian migrants years ago when they were all coming in post things happening in their countries. So I really do have a great depth. I was very fortunate that Lale, when she decided that she was going to step aside, was looking for somebody to head the group. And she reached out to me, and I'm delighted that I was offered the position, several rounds of videos with the board. And on the 1st of January, I really took over and moved forward with the organization. Next slide. So I mentioned the ambassadors. You can see the nice young gentleman down below wearing an NDF t-shirt, very nice. The gentleman on the top left, different color t-shirt. This is a remarkable group of young people. Their prime focus is getting the next generation tested to make sure that they're not carriers for the disease. And they do very interesting programs. For example, they just finished a boxing evening. 60 people attended, a lot of outreach, a lot of, obviously you can see they like having fun. And they're a great nucleus for us because from that base, we're able to expand. And just as a matter of interest, the person who's chairing this year's gala, Sogol Aminipur, she came from the ambassador. She's still an ambassador. So she is the event chair for this year's gala. And that's how we source new talent. And hopefully they will soon, as individuals, move up onto the board and, you know, not take the place off, but join with some of the more senior people who've been on the board for some time. Next slide. As I mentioned a couple of our board members before, we're really, one of the things we really have as an organization is great patient advocacy people. So our patient advocacy program managers, we call them PAPMs, they're quite remarkable because they really cover the universe, well, the globe. If anybody gets in touch with us, one of the four of them will reach out, talk to that person. It's usually a patient or a family member of a patient. Talk to them, give them some advice. It may be some advice on equipment. It may be some general advice on medications or something. But we're really fortunate to have these ladies, two in the United States, Mona in the UK and Maya in Israel. And they lead our group in really being the forefront of our patient-oriented programming. Next slide. Underneath that wonderful group, we have a group of 23 certified patient advocates in countries all over the world. What do they do? Well, they're again an amazing resource to patients, caregivers, individuals, medical professionals, scientists all over the world. And that was the first page. Here's the second page. So we got 23 in total. And that to me is a remarkable outreach. It enables us to be constantly in contact with patients. We have what are called huddles. it's an anagram for something. I can't quite remember what it is, but the huddle is the more important thing, where patients and patient advocates on separate calls get together. I've been involved in the two that have taken place since I joined, and it's a confidential outreach program where people can talk about anything and nothing that is of interest to them. And it again enforces that this is a global organization, and we really have a great opportunity to expand the base of the organization. Next slide. So this is something in which I'm very proud. And I invite everybody to go to the NDF website. It will take you hours to get through it. It's fabulous. And if you don't speak English, look at the languages you can now translate the website into. To me, that's one of the outreaches. I keep talking global. I keep saying what an amazing organization this is. But now if you live in Spain or in Turkey or in an Arab country or in India, several dialects, several languages in India are on there. It allows you to go and look, and we've got libraries of information. This webinar will go on the website, as have all the previous webinars. The information is open. It's there for the public. We can't tell you everything by sending you an email, but we can tell you everything by you going to the website, having a look, feeling comfortable, wiggling around. There's a contact us if you need to talk to us. So you have an easy way of really finding more about the organization. Transparency is what I bring to the organization. One of my mantras that I've learned over the years in nonprofit work, if I'm spending $1 of the organization's money, my first thought is, oh, that was money given to us by a patient. Would that patient be happy if I was spending that money? So if I'm ever invited out for lunch by a donor, I can promise you one thing: I'm not paying. I'm going to say to the, I would repeat what I always say to, and what I just said. I hope you don't mind if I don't pay, because if I'm paying without another donor's money. And that's not really why that donor gave us that money. So I love invitations to lunch. I love going out, but it's always on the precondition that somebody else is paying. That's just a personal thing. I think it makes a lot of sense. So here are the countries around the world where we have a presence. We're 45 and adding. I'm amazed by this. When I first came to the organization 3 months ago, I had an impression that this was a Persian Jewish disease. Well, it was when it started 2,500 years ago. But as you can clearly see, it isn't. And those are the countries around the world where we have a presence. I was talking to a wonderful gentleman from India one day, and we were talking about things, and he said, oh yes, I'm sort of multi-generation from India, but I have the Bulgarian variant. And that led me to think, well, how on earth did he get that? And of course, then we started tracing back. We got some great input from our scientists. And actually, it's one of our ongoing programs is to identify the variants and really help people who now know they've got that variant understand where it came from. So again, it's global, 45 countries and counting. We're going to expand this dramatically. The Soviet Union is a very large piece of all that. And as you can see, we're strong in Europe and everywhere else. The other amazing thing about the organization is we have a tremendous scientific advisory committee. There you can see the 11 members. And just to talk about the entities that they represent, everything from Hadassah University in Israel, Ohio State, UCLA, an institution at both university in Berlin, a second university in Berlin. We're delighted because Angela Leck, who's VP of Research at the Muscular Dystrophy Association, from whom you'll hear later, is really a wonderful advisor to us, and we're so proud to have her aboard. Monkolek, who happens to be her husband, is at Yale. As I mentioned, Tassin Mozaffar is at UC Irvine. Stella Mitran Rosenbaum is at Hebrew University in Israel. And it was she that discovered the gene, I believe it's now 21 years ago. We have Laura from the Jane Foundation. And last but by no means least, Perry Schill from UCLA Medical Center. So that's a remarkable body of scientific advisors. What do? They advise us on everything. Grants that come in, they advise us on those. The progress of grants and reporting on grants, they advise us on those. So this is one of the strengths of the organization. These are known as worldwide experts in the field, and we're honored and delighted to have them aboard on our scientific advisory committee. Next slide. So here's the important thing, and this is where I believe in complete transparency. And I never want a donor to say, where are you spending your money? Or where are you spending our money or my money? So here's where we go. Next slide. So we charted every year since we started and the grants that we gave. You can see 2007, a total of 270,000. 2008, total of 300,000, and the various entities to which we gave grants. 2009, 200,000. 2010, 295,000. 2011, next page. You can see the figure there. 2013, again, you can see the figure there. 2014, 164,000. 2015, 290,000. 2016, 190,000. 2017, 360,000. 2018, next page. And you can see the figure there. Look, you don't have to be a genius to add these together, but if you look at 2020, nearly 800,000. 2021, 485,000. So we have, in direct grants alone, it's almost $5 million. That's a very significant amount. It, I believe, exceeds anything that has been devoted to GNEM myopathy from anywhere else. And we're very proud of that figure. That grant information, as you can see, we have named individuals. They're all checked and researched by our scientific advisory committee and by our consultants in the field. We make sure that these grants are being properly monitored, and we obviously look at the results. The bad thing about science, it takes its own time. You spend a lot of time developing things. A lot of mouse models are what we have developed, and it takes time. They get from mouse models. We've got a whole series of things, and you'll hear from Dr. Leck in a few minutes. We're doing a great deal in the field, and it's all research, but as I said, it just takes time. Next slide. So that's where we have been spending our grant money. This is a very important document, and it's only an extract because it makes it pretty hard to show the whole document. Every year, every nonprofit has to show and file a thing called their 990. The latest one that we filed is for the year 2020. This is public information. You can simply go to it by Googling 990, check the name of the organization. If you're a member of GuideStar or one of those, you can check it there. But it is a forward-facing document, publicly accessible. Every nitty-gritty of the organization is there. I encourage you each to go and look at them. They're really important. So this is an extract from the 2021, which is the latest one that has been filed. 2021 will be filed over the next few months. It takes a little while to compile it. It's part of the financials of the organization. It's actually filed with the assistance of our outside accounting firm and auditors. So these are very carefully compiled figures. They go out under the name of one of our co-chairs, Ralph Lauren, and that's really important that these are forward-facing, transparent, and full of information. So if you will glance at line 8, you will see that in the year 2019, we got in $2.5 million. How did we do that? Well, primarily from grants, a matching grant, and from a gala. That was the last gala that we held. So our total revenue on line 12, 2.6 million. The current year, 2020, the income decreased dramatically. Primary reason, no gala. And obviously less money coming in from grants. And that was, part of the income shortage. But we still, on line 13, got 565,000 in grant money. So that was an important factor. Lower income, but continuing grant income coming in. And if you look at line 18, you see in the year 2020, we spent $1.3 million on things, and it shows a paper loss. It's only a paper loss of $900,000, but we managed to do all our research, keep everything going, get everybody paid, and that was the end of the year. It looks worse on paper than it is in practical terms, because now, next slide, please. So for the year 2021, and these are absolutely unaudited, this came straight out of our QuickBooks system, and it hasn't been checked by the auditors. We've barely finished 2021, but this is not a public-facing document, but it's the kind of document that I want individuals who are part of the organization to see and understand, because I don't want there to be any misunderstandings about income and expenditure. So if you glance on the left-hand side at the revenue, and you go down to total revenue, you'll see that the revenue is 436,000 odd, and that's not dissimilar to the amount that we had in revenue from the preceding year. You will see that we spent, and I'm only going down to the sort of the larger items, towards the bottom of the page, 4 items up, we spent $413,000 on research. And you can see that other than that, in that first column, there's not a great deal of expense. If you go to the right-hand column, you can see we have significant payroll expenses. We have professional and contract services. We pay stipends, smallish amounts, as you can see. God bless our panels, because they get paid a very small amount for the valuable work that they do. And again, in the year 2021, we're showing a paper loss of $715,000, but we kept our research going and we kept everything going as we wanted to. Next slide. So when I talk about paper loss, that's really what you have to think about. We spent more money than we got in, but we were able to maintain our financial position. So this is the balance sheet, again, unaudited, straight off QuickBooks. It may change, but it'll be minimal change. The most important figure there is about 8 lines down under other current assets. Line 132, Schwab investment, $1.2 million. We are economically sound. We have, at the 31st of December, $1.2 million in the bank. And obviously that is a designated amount. It is restricted to research. And that's our intention, to dedicate it to research. I will tell you now, as a group, there are 2 professionals working for the organization. You've heard from Rachel, you've heard from me. We're it. We don't have any office space. We both work remotely. so that when you see the 2022 financials, and I hope to have this call every quarter, you will see that in 2022, we've already cut overhead in terms of payroll significantly. We obviously have consultants who work with us, and they will be ongoing, but our overhead has decreased dramatically, and that's part of the forward movement of the organization. So you can see we got $1.2 million in the bank. If anybody knows how to read a balance sheet, the important thing is that total assets and total liabilities match, and God bless them, they do. It records just near the bottom that last year, yes, we showed on paper a loss of $715,000, but that's only paper. And you can see that other than that, it's a very clean and neat balance sheet and reflects extremely well on the organization, fiscally sound, continuing our ongoing research, and I'm happy to and proud to be part of the organization. Next slide. So one of the things that really matters to me is, where do we rate in the nonprofit world? For those of you who know, there's an organization that used to be called GuideStar, and this year, I believe, was acquired by a company called Candid, but we have maintained a platinum transparency GuideStar status for several years. The last recorded old-fashioned GuideStar was 2021, and we were absolutely top of the pile, platinum top-level status. And for 2022, already we are in that platinum level. I much prefer the old GuideStar logo. I hate the new Candid logo, and we're going to try and encourage them to be a little more GuideStar-like. But at the bottom of the page, I think the most important statement about the organization. Over the years, we have provided close to $10 million in funding for critical research, for symposia, and other programs, all within the goal of finding a treatment to end GNEM. I really feel that we have done what we set out to do. Have we achieved the goal yet? No. Are we working on a daily basis? Absolutely. Do we have great worldwide scientific research carrying on? Absolutely. And that leads me to the next slide. So I'm delighted to introduce a video by Dr. Angela Leck. As you can see, she is with the Muscular Dystrophy Association, the MDA. She's also a valued, she's their VP of Research, and she's a more than valued scientific advisor to our organization. So I'm now going to be quiet, and I'm going to allow the video to start.
Hi there. Over the past year, NDF has focused a lot of their effort and resources towards sponsoring an IND, or what's known as Investigational New Drug Application for GNE gene therapy. The process of proposing new drugs to be used on humans typically undergoes strict examination by the FDA, also known as the Food and Drug Administration. This is an exciting, new and focused effort which the NDF is calling the International Gene Therapy Development Program, or the IGTDP. Listed here are specific programs funded by NDF towards the goal of delivering gene therapy to GNEM patients in the clinic. Gene replacement therapy is a promising avenue to treat GNEM by introducing a new healthy copy of the gene packaged inside a viral vector known as AAV. This approach has shown benefit in childhood disorders such as spinal muscular atrophy and Duchenne muscular dystrophy, but has never been attempted for adult onset genetic diseases. We anticipate that this approach will result in clinical benefit for all GNEM patients, regardless of mutation type. In order for such a therapy to be approved for use in humans, there are a set of steps defined by the FDA that we must undertake in order to put together an IND package. Importantly, this project is led by NDF and not one individual scientist. This is a new and different model that we believe will increase our chances of success because it lets us tap into the multiple expertise of different scientists that we have funded for many years. Instead of one investigator, clinician, or scientist leading the effort and assembling their own IND, our NDF-led effort means we can better control the timelines and take a more proactive approach to pursuing gene therapy for GNE patients that is not solely dependent on the involvement of just one person. A successful IND package for gene therapy relies on the roles of several key personnel. Over the past year, NDF has assembled a team composed of these key personnel towards the completion of the IND package. With the NDF at the helm, we can now tap into individual expertise of each lab and compile the necessary preclinical data for an IND package for submission to the FDA. We have already completed the gene therapy design and are now actively testing 2 candidate constructs at Charles River Labs in a GNE mouse model developed by Dr. Mitrani Rosenbaum to determine the most efficacious and safest one, and also what dose to use in humans. There are a lot of moving pieces in this project, and we are consulting with Rich Horgan, our project manager, who has the expertise in managing a gene therapy project and IND submission to help make sure we are on track. With Rich's help, we have also obtained quotes from AAV manufacturing companies in preparation for generating a small batch of virus to deliver our candidate GNE gene therapy into a small group of patients. Our initial goal is to perform a small clinical trial with Dr. Tahsin Mozaffar at UC Irvine, who has seen many GNEM patients in the clinic. However, our goal does not stop there. The NDF feels an obligation to make sure that this therapy is available to all GNEM patients. The only way to achieve this is if we partner with a biopharmaceutical company with the know-how and budget to mass produce the gene therapy product. The NDF has been hard at work to achieve this goal and believe we are closer than ever to achieving this. This is perhaps the single most important goal that the community should get behind if we are to succeed in widely disseminating the therapy. In addition to this effort, we are also funding other scientists who are working with us to develop assays or readouts that are needed for the IND application to the FDA. For example, we funded Dr. Hawk and Horst Kotter to work on enzymatic assays for our gene therapy constructs to measure how a new GNE gene, once introduced into cells or mice, can restore enzyme function of GNE to catalyze sialic acid production. We also funded Dr. Martin to develop a potency assay, which is required by FDA to measure if production lots of gene therapy constructs works as they should before introducing them into humans. We funded Dr. Crow at Mount St. Joseph to screen a panel of lectins to identify better biomarkers that will tell us if a gene therapy has worked in humans. Finally, we funded Dr. Rosenbaum, in addition to Dr. Martin, this past year to work on developing a different type of GNE mouse model that will help fast track our gene therapy IND application. Current available mouse models of GNE are not ideal in testing gene therapy constructs, as they do not mirror the human GNE disease well. We therefore funded 2 groups to develop conditional GNE knockout models of the muscle that will hopefully be a better reflection of the disease that we can then use to demonstrate functional correction with our gene therapy product. These are just some of the projects that we have funded that all tie in together into a big picture of submitting an IND application for gene therapy approval by the FDA. Although the past 2 years of the pandemic have introduced many challenges, the NDF has continued to power through, and we are more focused than ever to delivering a gene therapy for GNEM patients around the world. We cannot do this alone, and we need the help of the entire patient community to get behind us and support our goal of ultimately finding therapy for GNE myopathy. Thank you.
It's me again. I think this is the ideal opportunity to reintroduce Dr. Leck, Dr. Weisleder, and Rich Horgan.
So we're eagerly awaiting the new phenotype And we're in touch with Stella's group, and she keeps us updated. Right now, there's still, this is the first generation of the mice that they're testing, and they're in the process of aging them out and trying to measure different properties of the muscle to see if there is any phenotype that closely resembles the human condition. But it's still really early to tell. I think give it a few months, and she should have more solid results by then, but she doesn't know yet.
Another question in the chat box this time. I'll read it out since I'm here. Just checking on what happened with the PerkinElmer study. Will the results be communicated for people who were tested?
Certainly know it's ongoing, and I know we're in touch with them. There's still some work to be done, but we're monitoring it on a weekly basis. I think Rachel was the last person to have direct touch with them, but it's definitely ongoing, no question.
There's another question here. Do you happen to know any updates on the Manic Trial as to when that will start?
I'm delighted to say that the Manic Trial has started. We obviously on the peripheral, we're helping, we're available to help recruit patient-wise, but it is definitely started. I think they're aiming at 15 patients in the first cohort, and it'll be weeks rather than months before we get any activity. In fact, one of the things I'll be doing, and I believe Mike is on the call, but I don't know. I'm in touch with the CEO of the organization that's running the trial, and we'll keep everybody updated.
Looks like there's another question. For my general information, what's the minimum data required by FDA to get approval for clinical trials? Can take that one. So the short answer is it depends. It depends on the disease, on the severity of disease in terms of how life-threatening or debilitating it is. And it also depends on the type of approach we're using. So whether that's a pill or a gene therapy can have significantly different ramifications. But in short, as it relates to this project, what we think the FDA will want to see is, of course, above all, safety. The FDA nowadays, as it's looking at gene therapies, requiring at least one animal model to be used, whether that's a small rodent model or a larger animal like a pig or a non-human primate. For our project, we think it'll be sufficient to use rodent models. And then also the FDA requires a burden of efficacy. So can we convince ourselves that we see some sort of data here that we think could result in a clinical improvement for patients? And so that's where we'll also glean from some of the in vitro or cell work, as well as mouse work or in vivo work that we're doing as well. But you essentially put together what's called an IND. This is an investigational new drug application, which includes 5 sections or modules. Each module goes into a different aspect of the program, whether that's the clinical aspect, what you'll do to measure patient efficacy and safety and monitoring. The next module is manufacturing. And then there's a non-clinical module as well, which is preclinical work, what we're talking about here with mice and such. The other 2 modules are less sort of experimental driven and more description of analyses worked on. Happy to talk more about that. And of course, if my other colleagues have anything to add, defer to them as well. Looks like
I think that's a good summary.
Questions. Oh, sorry, no, I didn't mean to go.
No, just go ahead. Go ahead.
So the next question, I'll just read it. I don't have an answer for this. Is another question about the Manic trial. Is it still recruiting or still ongoing? Can patients outside the US participate in it? I'm not sure if there's a clinical trial identifier that might be the most useful thing to provide to the audience here if somebody has that.
Yeah, there is. Geoffrey, I think Geoffrey would know the answer to that one, because I'm not very clued in. We're not very clued into the Manic stuff.
Joe, I can tell you that obviously it's ongoing, and at the moment it's only a US trial. I know that, and you can go to clinicalgov.org. Oh,
Clinical trial.
Thank you. Too many words. Thank you. There it is. And you can easily check, and it lists every clinical trial happening everywhere. So you can follow the progress and see where it is. And that's an easy answer. Oh, and I believe Tara wants to answer the question as well. So Tara.
My understanding is that the NIH will accept international patients for the Manic Trial.
Do they have to be in the United States?
No.
Okay. Thank you.
Okay. So Mangesh has a question. Can the panel talk more about the expected timelines for gene therapy IND application? Can you also talk about the challenges we still face? So regarding timelines, again, there is so many unexpected, I guess, hurdles that it's so hard to put an exact timeline. So it all depends on when we go meet with the FDA for the first time, what their response will be. You know, if they're happy with our plans, or they want us to do, if they want us to do like studies in non-human primates, for example, if they were to say, we want you to test your gene therapy on monkeys, that would drastically increase the timeline for the IND, and also increase the costs. But then if they don't require that, then that would obviously accelerate the whole process. It also depends if the IND is going to be like a really small clinical trial or a bigger clinical trial, say if it was sponsored by a company. The bar is a lot lower if we are to do a smaller clinical trial than if we want to get approval for a drug to treat a bigger patient population in the clinic, funded by a company. Does anyone, Noah and Rich, do you have anything to add?
Yeah, I think that's a great perspective that Angela's provided. I echo the comments around the importance of communicating early with the FDA so that we can get what their expectations are painted very clearly onto the roadmap. For those of you who have joined previous webinars we've done, the term pre-IND is something that comes up often. So this is a pre sort of application where you can get the FDA's feedback on the work that's been completed. So this would include the mouse studies, the design of the drug, as well as the proposed work to be conducted leading into the trial. And it's a really pivotal point because the FDA is less likely than sort of average to sort of pop surprises down the road when an IND would be submitted in theory.
So in terms of challenges, sorry, Noah, did you have something?
Yeah, I was just going to say that there's also some advantages to the fact with the Manic trial that's already been approved. It gives some indications about what potential endpoints might be as a trial is being designed. So that's certainly helpful. But as Rich and Angela both mentioned, the fact that we're layering a gene therapy on top of this, that creates specific challenges that have to be addressed in terms of safety and efficacy. But at least we have a bit of a roadmap that'll help us to be able to determine the best way to design the trial.
Yeah, I mean, I just want to again point out that systemic AAV delivery using for gene therapy, AAV based in a full-grown adult really hasn't been widely employed and approved by the FDA. All of the indications that have been approved are childhood onset diseases. It's really, really different to treat a full-grown adult with the enough virus for all the muscles to be treated. So you know, I can foresee the FDA having those concerns about the safety. And in terms of, can you talk about the challenges? I mean, obviously money is, funding is an issue to, you know, and what else? The mouse model not having a phenotype is another issue. Having a robust readout of if the gene therapy were to be dosed, what can we measure to see if it's been successful? Because it's such a slowly progressing disease, GNE, we won't know straight away if it's worked. Because, you know, it's different if we want to measure a decrease in progression of the disease as opposed to completely curing the disease. It's like two different questions. So that's also another, how do we structure the clinical trial so that we'll know if it's been successful or not? That's a huge one.
Yeah, the other side of that, looking at the challenge as it relates to dosing adult patients with AAV. So, I mean, that's clearly a significant challenge going forward, simply because it hasn't been done extensively yet, and because the FDA hasn't approved these sort of trials. So that makes the studies looking at the safety of this approach particularly important, and something that the FDA is going to want to address robustly. I would say that also there are advantages to the current state of the technology. When we look at the technologies that are in clinical trials now that are dosing younger patients, and now some early trials looking at adolescent patients, the challenge there is you're using older technologies that have taken this time to develop through the clinical ladder that we already discussed. And so you're using technologies that were state of the art maybe 5, maybe even 10 years ago, depending on the particular technology. Because of all the interest in gene therapy, which I'm sure everyone on this call is familiar with, there's been a great investment by a lot of different laboratories and a lot of different companies to come up with new technologies, particularly to target muscle, to develop new types of AAV that are more efficient at transducing muscle, and also new ways of driving the expression of the gene that you're going to replace. So all of these technologies that are coming on board now, that are coming online, give us an advantage as we start to put these together. And quite frankly, will probably lead to a better, even looking now, than we would have been able to put together even 2 years ago. So one of the great advantages of where we stand now is the fact that we can bring these newer technologies on board and make it more likely we can effectively dose adult patients in the future.
Noah, you're cutting out on us.
Let me take over, because I hope everybody saw Michael's.
That's probably why Wi-Fi.
Michael's posted, and Michael is the CEO of the company that's running the Manic Trial. His words are, Manic Trial has begun at NIH and Brigham and Women's with patient enrollment this month, with other sites being rapidly opened in April. That's a very positive sign, and thank you, Michael, for confirming that. I see there was one other question I thought I saw from.
3 questions.
Thank you. Go ahead.
What is the expected outcome of gene therapy? Is it expected to reverse muscle wasting or stop the muscle wasting? So again, this is really unexplored territory, but if I had to guess, being really conservative, I would say it's to stop the progression of the muscle wasting. Reversing the muscle wasting is a completely different ball game, because when you have muscle wasting, you get fibrotic tissue, and you get infiltration of fat tissue that you can see by MRI in the space where your muscles are supposed to be. It's really hard to get rid of that and to change that back into muscle. I would say in the future, the technology may be there to reverse the disease, but I think for now, gene therapy would be the aim of gene therapy is to stop muscle wasting. So Sudha has a comment: due to challenges of AAV-based gene delivery in adults, are you considering delivery directly to muscle rather than intravenous? I think that the appetite for intramuscular delivery is very low, because people only get one shot at gene therapy. And you want it to go to as many places, as many muscles as you can with the one hit. If you just deliver it directly, I think that it's a wasted opportunity. There have been trials that the initial trials of gene therapy did only do intramuscular, but again, then you're targeting just like one muscle group, and it's just not very hopeful for the patients to just sort of correct the mutation in a localized area.
Tara has a question. Can you address gene replacement, naked DNA, plasmid delivery versus AAV? So gene replacement is the same as AAV, because gene replacement, you need to put the gene inside a delivery vehicle. So the AAV serves as that delivery vehicle. There are many other types of delivery vehicles that don't involve AAV, which is a virus, such as nanoparticles or lipid particles. Those are currently being trialed in other diseases, but they're not proving to be as efficacious. They don't get to the muscles as efficiently as viruses, because viruses have had like millions of years to evolve to transduce, to infect human cells to get their payload inside. So that's why scientists lean heavily on viral-based delivery vehicles. In terms of naked DNA plasmid, that just doesn't work. People have tried that in the very early days of gene therapy to do that. It really just doesn't work. Yeah, that's my take. Noah, did you want to say something about that?
Yeah. So that's something that's been explored for years in the field, right? Through various ways of delivering it, hydrodynamic delivery, through the application of, which is basically swelling the muscle with fluid and then like letting it shrink back down, with electroporation, other types of direct injection approaches. The challenge with the challenge with any time you're using plasmid DNA is it doesn't have that viral mechanism around it. Like as Angela was saying, viruses are great at getting DNA into cells. That's what they do. And in order to get other ways to get it into the cell, you have to disrupt the membrane in some way, whether it's by swelling the cell or by using electrical shock on the cells. There's a lot of different ways to do that. But you inherently damage the muscle doing it that way. And the challenge when it comes to neuromuscular patients is you already have difficulty with the muscles. And when you damage them more, that can lead to longer term effects. It's also, we talked about earlier about intramuscular injection. A lot of those sort of approaches require injection into all the affected muscles. And then GNE, that's most of the muscles in the body.
That's a lot of.
So you're talking, in some cases, hundreds of injections that you'd have to do in order to make that happen. And those themselves cause difficulty and injury as well. So there's a reason that the field has coalesced around using these AAVs to deliver this approach, to deliver these sort of gene therapies. And it's because it allows you to avoid a lot of those potential complications with these older technologies.
So yeah, look, I just want to add, like if delivery of DNA, of GNE, or any gene were to work, then there would be no need to package them into anything, into any delivery vehicle. But the fact is that everyone is investing millions, billions of dollars into these delivery vehicles, because the fact is you can't get your muscles or any cells in the body to just take up naked DNA. It really doesn't work. If it does, then people wouldn't be trying to cleverly get around it using all these methods.
Yeah, the manufacturing of AAV is extremely complicated, and it's one of the things that's been a real challenge for the field to develop these sort of gene therapies. Thus, because of that if there were another way to do this that would work effectively, I assure you that the Mercks and the Pfizers of the world that really want to do these gene therapies would be pursuing those aggressively. Because all of those, it's much easier to make plasmid DNA. It is almost any biomedical laboratory in the world can make plasmid DNA effectively. The reason that AAV is being used is because it's really currently the gold standard and really the only practical approach to be able to deliver that at a systemic level throughout the whole body and affect all of the muscles. There's other things on the horizon. There's other things that could potentially do that, but that is literally decades away in all likelihood. When I was in graduate school, which is longer ago than I like to admit, that was when AAV first came online, and it's taken almost 20 years to be able to get that from the idea phase to the actual implementation in human clinical trials. As I mentioned earlier, the newer technologies that are available have certainly accelerated that, and we see more and more trials coming now. So that's an advantage that takes place now that didn't exist 20 years ago, because now these roadmaps and these new technologies. But quite frankly, that's the challenge here, is to make sure that we can move this as quickly as possible, but in a way that's going to be fully effective and safe for the muscles that are going to be transduced.
Let me
yeah.
just tell you that I'm afraid we're now at the sort of the hour mark. We said we'd try and conclude this in an hour. One of the things that I haven't mentioned is some of the issues that are being addressed. For example, we are talking to a biopharma company about some opportunities that exist, which would benefit both NDF and the patient population, but it's too early to talk about that. But we hope that by the time we get to the next quarter, or there may be a press release earlier than that, just informing everybody what's going on. I would invite anybody who didn't get a question answered to contact us through our contact us address at NDF. In the meantime, I really do want to thank those who participated in this event. Our panelists, so appreciated. I love working with each of you. I particularly like working with Angela because I spent many years in Australia, in Sydney, so we have a great background in common. And to Noah and Rich, thank you again both. To all participants, thank you so much. And I hope everybody has a nice Sunday, or if it's already Monday where you are, a pleasant week. Thank you.